Okay, let's keep going. So in the eyeball, the most important area, that's the retina. Retina is this piece of paper. And you have two different kind of receptors, the rods and the cones. Let's look at the rods and cones. So that's the rods and that's the cones. The cones, we have three different kinds of cones. The green cone, the blue cone, and the red cone. So the cones are for color vision, and they give you very sharp vision. But these are the expensive toy, so we could not afford too many of them. We have those cones, a lot of them put in the fovea area. And they need more light to activate them. And we use them for the daytime vision. So during the daytime, we have plenty of light. We have very acute vision. That's the cone. And the rods are the cheap toys. You only need a very little light to activate them. So they are perfect for the night vision. But you only have one kind of rod, so no matter what wavelengths you hit it, it gives you exactly the same response. And that's why the rods are for night vision. And during the night, you don't see color pretty well because the rods is for black and white. And on your retina, you have this too. So people like to use a camera to represent your eyes and the film. Well, we don't use the film anymore, but we used to. This is my first digital camera, the film camera. And they say the eye is like the film, but actually the retina is a smart film. It's not just a film. So let's look at the retina distribution. And we have the rods and cones. And the cones are the better one, sharp vision. But you could not afford to make the whole film that expensive. So you put those cones, three kinds of cones in the area, very center area, is called the fovea. So if you look at the distribution of the cones, the red one, you found that's the fovea area. There is almost all cones, no rods, all cones. And when you start to walk away from the fovea, the number of cones significantly drop, and the number of rods increase. So the fovea, that's the area with good visual acuity, means those high resolution sharp vision. That's the cones. That's the optic disc. That's the area your nerve and blood vessel goes through. So this part is, there's no photoreceptor. Actually, there's a blind spot, and then you start to go back to the same, right? So the fovea area, that's the one with three kind of cones, and the peripheral you have rods. And the visual pathway, the signals, the light hit the retina, and the retina gonna transfer the light into the neurotransmitter release, then the action potential, and the action potential will go to thalamus. This area is the thalamus. So in the chapter 3, we say the thalamus is like a train station, right? Information move in, move out of the brain. That's the thalamus. From the thalamus, it will go to the occipital lobe, and that's your primary visual cortex. So if it will go through your cranial nerve number 2 and go to your V1. V1 is primary visual cortex. And let's look at the color vision. The color vision is a privilege for human. If you don't have color, you can survive. It's not so convenient, like these flowers that are pretty. We take the color out, well, they're not so pretty, but you can still see that they're flowers. So some animals, they don't have the privilege, they don't have color vision, and they still survive. But if you see color, it's convenient. Like I put a strawberry, a red dot somewhere. If you don't see color, it's not so easy to find the strawberry. But if you see the color, it's pretty easy to identify it. So that's the privilege. That's why we have the color vision. Sometimes it's easier to identify the food, and the rotten food, it's important to have the color. And to talk about the color, let's talk a little bit of this person. Yeah, Newton. Newton is a physicist, but he also studied the visual system a lot. He invented this. So he's able to separate the light into different spectrum. And Newton is pretty smart, and he studied the light, the visual system. Eventually, do you know what's his conclusion? His conclusion is your color perception is based on your neuronal activity, based on your brain. It's not based on the light. That's pretty amazing. That's why he's the smartest person. He's not one of us. He's too smart. So let's look at the color vision. We have color because we have three kinds of cones. We have three kinds of cones, and the blue cone, the green cone, and the red cone. The red cone is actually not red. If we look at the wavelength, it's close to the yellow range, but we like to call it the red cone. And you are able to create a different color. It's like different percentage. It's like if you learn painting, you found your teacher will show you. You can use the blue ink, the yellow ink, and the red ink, create the whole spectrum. And why? Because that's how your visual system work. So you take a little bit blue, a little bit red together, and you create the purple color. And that's how you mix these two together. And if you take the green and the red together, you create orange. And that's how they create the whole spectrum, because that's how your visual system work. And if you have more cones, your world will be prettier, like this guy, the mantis shrimp. This guy have 12 cones. I don't know why he need 12 different kind of cones. His world must be beautiful. How beautiful? I don't know. I only have 3 kind of cones, and we never experience it, so we never know what we miss. And this human's cone, we have 3 different kind of cones. Good enough. And this guy has 12. So start from the UV, right? Different kind of UV, and start from purple, the blue, the light blue, and yellow, and wonderful Longhorn color. We have to use the red and the green create Longhorn color. Here's wonderful Longhorn color, and the red to the infrared range. His world must be beautiful. How beautiful? I don't know. I never experienced it. And we're good with 3 kind of cones. Sometimes if you can see UV is convenient. Like I give you the real money with counterfeit money. They look so similar. The reason is the spectrum of these two, they are almost identical in the visible light range. We talk about the visible light from 400 nanometer to 700 nanometer. They start to separate in the infrared range, and that's how they check, oh, this is the counterfeit one. If we are able to see the infrared, it would be convenient, we can see it, but we could not. And that's another example. Do you see a tank here? No, impossible for me to see. Do you know why the tank have to paint with this kind of color? And the reason is camouflage. So they have to use the infrared camera to take a picture. And the spectrum of the tank and the background totally identical from the visible range from 400 nanometer to 700 nanometer. So that's why you see no tank here. And they start to separate in the infrared range because the engine creates heat, and heat is infrared. And turn out they have to use the infrared camera and take a picture and overlap with the picture, and found the tank is here. And that's an experiment you can do at home to show you you actually have 2 different systems, photoreceptor system, the rods and cones. And the cones are for color vision, but they need more light to activate them, which means you are able to turn off the light until the light only activates the rod system and not the cone system. And suddenly the red flower turn into a black and white. Or you can use your shirt to do the experiment. Actually, I accidentally do the experiment. It need to be, based on my experience, this needs to be very, very dark. So you have to turn off all the light, and at night, you could not have a little bit of light bulb. It's too light. How I did this experiment is in the morning, in the winter, I put on the shirt. I thought I put on the black one until I went to the bathroom. I turned on the light. I found, oh, okay, I actually wear a red one. And that's exactly this, because you have the rods and the cone system. And the rod system need a little bit of light to activate them, but they are for black and white only. And you need to have enough light to activate the cone system and to see the color. So you can turn off the light to dim enough to shut down your cone system, only have the rod system. And your color vision, because you have 3 different kinds of cones, so it's called the trichromatic theory. It's based on these 3 colors, and you mix them. And also, when they process the color, it's called the, next slide, the opponent process theory. So your red and green being processed together, your yellow blue being processed together, and the third group is black and white. So it turned out when they process the color, these two being processed together, so you can make your neurons tired of this one, and suddenly you see the green one. That's this experiment. Let's do this experiment. I like to do experiment. And I provided the link. There's a link. You can click this, or a YouTube video. You can watch this. Okay, let's do experiment. So you need to focus on here. That's where you focus, and use your peripheral vision. Look at this dot. And you found this dot disappear, right? Okay, not a big deal. Keep paying attention to this. After 15, 20, 30 seconds, you start to see the green dots. Do you see the green dot? If not, you have to pay attention to this. So this tells you, okay, there's no green dot there. It's your brain created. Why your brain created? Let's go back to this. And that's the opponent process theory. Because your brain processes these 2 colors together. And it's all based on neuronal activity, because your perception is all based on neuronal activity. So you have equally amount of neuronal activity of these 2, and they are balanced. And when I keep showing those red dots, and the neurons, at first they fire, and gradually they are tired. They fire less and less. When they fire less and less, even though the green, there's no increase in the green, but the red decrease. And when these 2 process together, when the red decrease, it's like the green increase. And that's why your brain see the green after image. And that's the opponent process theory. So in the dot, if I change to the yellow, you're going to see blue, because these 2 are processed together. The white and black processed together. So that's the opponent process theory. And your color vision is based on these 3 kinds of cones. And if you miss one kind of cones, you're colorblind. And in theory, people can miss any one of them, but most people miss the red one. So that's why the colorblind test, most of them are going to use the red background and use the green later to test you. Because most people, it's a genetic, and they miss the red cone. Or they don't have enough red cone. They don't need to completely miss it. They just don't have enough. If you can see 74, you're fine. If you're colorblind, it's not the end of the world. You still see most of the color. And this picture try to mimic what the colorblind people with missing red cone look at the picture like. So this is the picture with all the color component. And that's the colorblind people. They see the picture. This look like. You found they still see certain spectrum. It's like I give you only the green and the blue color, ask you to paint. Okay, you miss something. You miss something. You miss the red component. It's not the end of the world. And plus, you never know what you miss. So you still have your normal life. Pretty good. And eventually, the signal gonna go to your brain. You use your brain to see. So if you are interested, we can show you this. You can look at this illusion. Pretty amazing. Because your brain, they can create the image and make a 2-dimensional look like a three-dimensional image. Because your brain create the 3D. Okay, now the signal go to the brain. Let's look at the neurons in the brain, how they behave. This area, occipital lobe, this area is called the V1, primary visual cortex. Well, actually, let's take a break.